Hello researchers from all over the world. My name is Lizhi Liu. I'm currently studying at the Shanghai K Laboratory of Intelligent Information Processing at the School of Computer, Fudan University, engaged in bioinformatics research. It is my great honor to be here to introduce my paper, HPOLabeler, Improving Prediction of Human Protein-Phenotype Associations by Learning to Rank. Xiaodi Huang and Shanfeng Zhu are my co-workers. If you have any questions at the conference, please feel free to send an email to me. First of all, let me introduce our task. As shown in the left figure, given a novel protein, what we need to do is to predict the HPO annotations of it. The Human Phenotype Ontology (HPO) provides a standard vocabulary of the human abnormal phenotypes and of the semantic relationships. The structure is quite similar to the gene ontology. Here, we focus on the most important sub-ontology, phenotypic abnormality, or PA. It is particularly noteworthy that our problem is not to predict which protein is most likely related to a given HPO term. Our setting is protein-centric rather than term-centric. The formulations of these two problems are quite different in practice. In our paper, we proposed a novel method called HPOLabeler. The framework is shown in this figure. The highlights of our proposal are threefold. First, from a macro perspective, our model utilizes stacking in example learning. Second, multiple basic models are integrated by learning to rank to further improve the performance. Finally, our method is the only one with better performance than naive methods in temporal validation. Now let's dive into the model. At the beginning, we extract several features for these models. The first kind of features is generated from protein-protein interaction networks. Here we choose three PPI networks: string, Germania, and bigrid. The jth entry of the feature vector xi represents the interacting score between proteins pi and pj. Besides, we create feature vectors from HPO annotations. That is, for protein pi, we have three binary vectors, one for each sub-ontology. We also use Temporal scan to predict domains and functional size of input protein by Temporal predictive models. It has signatures. And then we get a binary feature vector with jth entry being 1 if protein pi is matched with signature j in Temporal. The last feature is generated from amino acid sequences. Here we count the frequencies of all possible streamers in the AA sequence, resulting in a feature vector with each entry being the frequency of the corresponding trigram. So far, we have generated a total of 8 different feature vectors. For protein pi and HPO term T, from feature vector xfi of data source F, the predicted score SFPIT is calculated by a logistic regression model. So we have 8 scores for each protein HPO term pair. According to previous research, two proteins with strong interactions are likely to be associated with same phenotype. The formulation is given here. The basic idea behind it is that the association score between protein Pi and term Tg is the weighted average interacting score of Pi's interacted proteins. In other words, the neighboring nodes in PPI network. In Kappa-2 challenge, naive methods shown impressive performance. So here we consider it as another basic model. We use a number of appearances of HPO term T as the score of all proteins for that term. That is, the score is always the same for any proteins. Now we have 12 components, but how can we integrate them properly to further improve the prediction performance? The answer is learning to rank. To this end, we need 3 steps. The first step is to generate the candidates. We sort HPO terms of each protein in descending order of the score from component models and select top K of terms as candidates. Then we union the candidates of each component as the final candidates. For each candidate HPO term, we concatenate the predicted scores from 12 models to generate a feature vector to be input to learning to rank. We call this representation as string of scores. Finally, we use a state-of-the-art learning to rank algorithm lambda-mart to re-rank the candidate HPO terms. By feeding the string of scores representation into model, we receive a ranked HPO term list with predicted scores, and these are the final prediction results. We first evaluate our model by cross-validation. We choose HPO annotations released by July 2018 as the dataset. Here are the results of each component. In our paper, Fmax is protein-centric metric, AUC is term-centric, and AUPR is pairwise metric. From the table, we can see that the nearest neighbor is the best basic model, and PPI is the most important data source. What's more, by comparing NN with LR, we can see that our proposed NN method is better to utilize PPI network. This table shows the impressive performance of HPOLabeler. Our model improves AUPR of phenostruct, one of the state-of-the-art methods, by nearly 20%. And HPOLabeler outperforms all the components, indicating the effectiveness of learning to rank. However, our methods not significantly outperform the competing methods. What's the reason behind that? In fact, The distributions of HPO terms and HPO annotations are quite unbalanced. In the left figure, most of HPO terms only annotate no more than 10 proteins. In other words, many of terms are low frequency, so we divide all HPO terms into 6 groups according to the number of appearances. The right figure shows the statistics of these 6 groups. Figure A is the proportion of HPO terms in each group, and Figure B is the proportion of HPO annotations related to the terms in each group. As can be seen from the figure, the HPO terms annotating only a few proteins account for the majority of the entire HPO. Here we list the AUCs of of 4 out of the 6 groups, indicating that HPOLabeler outperforms all other methods in high-frequency groups, but falls in low-frequency groups. This implies that the performance of HPOLabeler is superior for more common HPO terms. Furthermore, we conduct leave one source odd check by eliminating each data source from the learning to rank framework in order to find the most informative data source. The results shows that the performance of the model eliminating PPI is most reduced. Again, it indicates the power of PPI network. Besides, we can see the AM method is the most important basic model among them, and all chains are negative, which suggesting all components are indispensable. To further evaluate the performance of our methods, we conduct temporary validation here. The setting is similar to Kappa challenge. Here are the results. Our method is the only one that is naive. The result is consistent with Kappa 2. It is so strange, right? Naive doesn't use any data source but achieves good performance. We think one of the reasons is incomplete annotations. From Figure A, we see the newly added proteins in the test set have fewer annotations than old proteins. By using different annotations released at different times, we find that AUPRs are increasing. Thus, we can imagine that HPO terms annotated wrongly might be currently false positives by incomplete annotations and become positive by biological research development in the future. This table can support our claim. Many high-ranked predictions are wrongly regarded as false but actually positive. As time goes by, the number of HPO annotations of proteins continues to increase, so the incompletion does matter. We also developed an online platform. You can enter a protein ID or gene name and get a list of predicted annotations. In summary, we propose HPOLabeler by using learning to rank to integrate multiple data sources. The evaluation results show that our algorithm significantly outperforms competing methods. We find that PPI is the most informative data source, and lower performance in temporary validation might be caused by incomplete annotations of new proteins. And please enjoy it on our online platform. That's the end of my presentation. If you have any further questions, please feel free to ask me. Thank you for your listening.